So how do you figure out who should be treated with what drug? And in some cases, there are very clear markers that identify patients who would respond to a drug. Unfortunately, in many cases, there are not. And you can only test so many drugs in so many patients in vivo. So there's been a lot of interest in ex vivo predictive testing. Now, next speaker, Dr. Cyril runs the Center for Molecular Therapeutics at MGH and has been using both established cell lines and increasingly patient-derived cell lines to look for drug sensitivity. Cyril.
Thanks, Daniel. I guess you gave my talk already, so I'm good. Hi, everybody. Imagine that it's 1999, that Prince is still alive, and we haven't sequenced the human genome yet. Now come back to present day, and you realize how much genomics have changed the way we think about cancer and the way we think about establishing new therapeutics for cancer. But today I'm going to tell you something different. I'm going to tell you how we think about establishing a functional diagnostic for individualization of cancer care. This is a group of people at the Massachusetts General Cancer Center, mostly in the thoracic group that have worked on this, together with me and Jeff Engelman. So in the last 15 years, we've seen a big transformation in the way we think about establishing clinical trials and bringing cancer drugs to people. And that's based on the way we understand cancer and the way we can now very efficiently sequence the genome of cancer and bring this information to the clinic. It makes clinical trials smaller, more efficient, and it makes the whole process a lot cheaper. And it obviously has had tremendous effects, specifically in leukemia, with essentially the cure of chronic myelogenous leukemia, but also in solid tumors, both in melanoma or non-small cell lung cancers, with effort led at MGH by Alice Shaw and Nisha Sequist, for example. However, there are some problems with this plan. One is the enormous genetic heterogeneity of cancer. That makes it very difficult to imagine how we're going to test all these drugs or this drug combination in clinical trials. There's not enough patients, there's not enough money, there's not enough time. And there are too few cancers that are matched with targeted therapeutics. So we need to do something much quicker than that. And then even for the ones that work the best, acquired resistance, the effect that under the drug, initially there is response, and then it stops, and patients are sick again, acquired resistance limits dramatically the benefit or even the best therapeutic today. So a few years ago, with Jeff Engelman, who was working on acquired resistance for a while already, we figured that maybe what we should do was simply take cells from small biopsies, from fine needle biopsies, and try to figure out whether we could figure out which drug will work on these cancers again, making them sensitive to the initial therapy again in combination with a panel of new drugs that we'll be testing in the lab. And we knew this was potentially important because a lot of the time, the genomic information that you get out of comparing the initial tumor and the acquired resistant tumor is not good enough. You don't know what to do. You don't know what to do sometimes because the results are ambiguous. There is more than one mechanism that is suggested by the genomics. Or sometimes you don't know what to do because nothing that you have there you understand. And that obviously reminds us that we understand very little about cancer. So because we understand very little about cancer, we thought let's have a simpler plan and let's test drugs that are available in the clinic first and maybe in the future new drugs directly on these cancer cells. So we took these cancer cells, and it takes a few months at that stage to make a cancer cell lines, and then we try directly drug combination on these. And what we could see immediately is that when we add a culprit in the genome, it worked. The cancer drugs that we tried in the lab were the ones that were working according to the mechanism that the genome information was giving us. More importantly, when the results were ambiguous, it also was giving us which drug we should be using, meaning there were two potential mechanisms of resistance, but only one drug that was targeting one of these mechanisms was actually working in the cells, in the drug screen that we were doing in the lab. And then finally, obviously, the most interesting was that sometimes, like I said before, the genome was giving us absolutely no information, but yet we could find a drug that in combination with the initial therapeutics was killing the cancer cells. So that made us think what we need to do now is we need to broaden this and bring it not only to the acquired resistant context, but even to the first line. But we also need to make it faster so we can bring it to patients from which we take the biopsy, take that biopsy, test the cells in the lab, and go back into the clinic together with the genomic information and inform the care of the very patient that we take the cells from. So that's our plan. We're going to take these cells, and in 4 to 5 weeks, we're going to culture them, and we're going together with the genetic test, not to replace it, but to complement it, we're going to inform the care of these patients. Where are we now? What we know is that we can, in fact, culture cells from these fine needle aspirates, from small biopsies. We can take that, fine needle aspirates or core biopsies. We can take these cells and we can grow them. The problem is that it takes a few months right now, so we need to make it faster. The other thing we know we can do is that we can take cells in the mixture of what comes in the biopsy, stroma, fibroblasts, etc., and we can specifically look at the effect of the drugs on the cancer cells. So we know we can detect effects specifically on the cancer cells. So we're good for that, at least in lung. So what we need to do now is to make this faster. So we're working on developing new methodologies so that we can grow these cells again in 4 to 5 weeks and inform the care of these patients. Initially, what we're going to do is we're going to try only a few of the drugs that are available in the clinic. So we won't have to grow a ton of cells. It'll be fine to just have enough to try 10 to 12 drugs, maybe even less. And then we need to have a proof of concept, but we will show that, in fact, we can inform prospectively the effect of these drugs together with the genomics, going beyond the genomic information. And that's it. Thank you.